As we begin to look at each of the subsequent chapters, you're going to find that most of the topics that we talked about, with the exception of the last few chapters, were already covered in Chapter 1. Most of the topics that we will talk about were covered in Chapter 1. Some of the latter chapters talk about systems when the immune system fails or when it overreacts or aspects like that. And so those will relate back to the basic knowledge that you're going to gain. But most everything else was discussed at least at some level in Chapter 1. The subsequent chapters just give you more specific detail. And that's the case for Chapter 2. Chapter 2 is talking about the innate immunity or the innate side of immunity. And before we can talk about that, we have to think about the various types of pathogens that can come into our systems. Pathogens are all different. All organisms are different. And if you think about organisms that are out there, some have mechanisms to persist in the environment for very long, extended periods of time, like those organisms that make endospores. There's some evidence but scientists have dug into the core of the earth and found endospores that were buried whenever the earth was formed down in pockets below the surface of the earth. And they're still viable, and they revived them, and they look a lot like organisms that are up on the surface of the earth, but where they're discovered, there's no way they could have gotten there except for during the formation of the earth. So those guys have persisted for quite a long time in the environment. Other organisms or infectious agents don't persist nearly as long. An example we just talked about is the AIDS virus. It's got a very short seconds half-life out in the environment. For example, on a surface or on hands of someone or something like that. Its outer layer, it's an envelope virus, and it has a membrane on the outside, and that membrane is very labile or easily broken or destroyed, and it falls apart very, very quickly. So about the only way that HIV can be transmitted is through either bodily fluids or intimate contact with another individual, because when it's outside of the body, it just doesn't persist very long. And each organism has a different ability and a different half-life as it's persisting in the environment. I used to have in my lectures for the previous book, I had a nice scenario for half-lives to help you understand that. I was thinking as I was preparing for this lecture, I looked through, I didn't see a lot of you that listed biochemistry as a course that you've already taken. How many actually have taken biochemistry already? Yeah, that's what it looked like from the sheets. I didn't see very many. So if some of the things we're going to talk about in Chapter 2 relate to biochemistry, hopefully that'll help you prepare for that class down the road if you need to take it for your minor. But a lot of that has to do with stability of proteins. And I have this thing in the other lecture about Cookie Monster. And if you give Cookie Monster 50 cookies on day 1, and he eats half of his cookies in 24 hours, how many cookies does Cookie Monster have left after 24 hours? 25, beautiful. So the half-life for cookies with Cookie Monster, that may be exaggerated, and that scenario is 24 hours, right? And so after 3 days, how many cookies does Cookie Monster have? Yeah, 6 1/4, right? Because after day 2, there'd only be how many? 12 1/2, and then at day 3, there'd be half of that, so 6 1/4. And you can go on down, and theoretically, Cookie Monster would never run out of cookies, but by about day 7, he has less than 1 whole cookie, and Cookie Monster is probably not going to be too happy at that point. So I put that scenario into my lecture notes because a lot of students haven't had biochemistry, and they don't understand the process of half-lives. But we'll be talking about and using that term half-lives over the course of the semester related to different cells and how they persist and other organisms in the environment. Well, depending on the infectious agent, and I'll just use that as a general term to relate to bacteria, fungi, protozoan, viruses, and all the other types of things that can infect. Depending on them, they have different mechanisms to damage tissues in our body. Some of them release toxins that we call exotoxins. All right, you guys have all had microbiology. It's a prereq for this course. So what is an exotoxin? Somebody tell me quickly. What's an exotoxin?
Something that's released by the invading organism
Okay, I'll go with that. You could have been more specific because it's in the name. You could have said a toxin that is released or a molecule that's toxic that's released by the invading organism that can damage the tissues. And that's what we're looking at here. A lot of pathogenic organisms do make exotoxins. And one example that they list, and they just give a very limited example for each one, but that organism that's come in is releasing molecules, and usually exotoxins are proteins, and they go and bind to receptors on the cell and do something to those cells. Sometimes they disrupt membranes, but most of the time they get internalized, and down inside of the cell they interfere with some process down inside of the cell. It may be DNA replication, it may be some of the other nuclear functions, it may be RNA production, it may be a number of things depending on the particular exotoxin. Some of them impact epithelial cells, some of them impact endothelial cells, some of them impact specifically neural cells. It depends on the function of the given toxin that you're talking about. So you all remember E. coli, right? That's the guy that lives in your gut. You guys are healthy, no problem. But there's the cousin E. coli out there, that O157H7. It actually picked up some exotoxin genes from one of its cousins, Shigella, and when it expresses that toxin, it makes it much more pathogenic and more lethal. And so that's just one example that you guys know of. Okay, here's this cool one. It's called endotoxin. Somebody tell me the other name for endotoxin. It's got another name. And you should remember it from microbiology, because those of you that took my class actually had to label the various parts of endotoxin on the final exam, usually. I didn't ask you last semester if you were just in there last semester, but previous semesters I have asked. Yeah. Beautiful. So endotoxin specifically, lipid A is part of endotoxin. What's the other part that's connected to lipid A? She threw out a talking point. So what else is connected to lipid A? A long chain. What was it? It's a polysaccharide chain, so you're getting close. So it's got a long polysaccharide connected to lipid A. So what's the other name for endotoxin? LPS, beautiful, lipopolysaccharide. It sounds like I drew some blanks there. You guys need to go back and review microbiology and look at that structure. And the main reason I test that multiple times in micro is because that particular molecule is very immunogenic and very much a stimulus. In fact, we put it into some vaccines to make the vaccines stimulate a stronger response. So we'll purify LPS or endotoxin and put it in there. But LPS has its own properties, and it can kill cells through its structure. And you mentioned lipid A. Lipid A is the toxic part of LPS. The polysaccharide part isn't toxic, but LPS, depending on lipid A, depending on how the side chains are put on that lipid, make it more or less toxic. So just a refresher again, what kind of cells have endotoxin? What kind of cells make endotoxin? Gram-negative cells. And where are you going to find that? On those cells or in those cells? The outer membrane on gram-negative cells, and the outer layer of that outer membrane is going to be endotoxin or LPS. If this isn't clicking and ringing bells, then you definitely need to go back and look at some of those important structures that are related to microbes, because I don't have time to review all of those as we're going through, and I'm thinking if I say endotoxin or LPS, that brings up beautiful pictures in your mind, because I tried to burn them into your minds as you were taking microbiology. So you should be able to pull those up pretty quickly. If you're reading, which you should be, the chapters before, and you hit words like that don't bring up a beautiful image, have Wikipedia up on your computer. Go throw it in. It's a beautiful resource, even though some faculty don't like it. Pull it up. It's going to have pictures of all of those molecules and all of those things that we're talking about, and you can very quickly get a mental image of what's going on with those molecules. So that's endotoxin, but a lot of, we just said all gram-negatives make endotoxins. Some endotoxins are more toxic than others are. Okay, and then there's direct cytopathic effect, and that's the effect that viruses can have. They get into the cell and they kill the cell, and we didn't get very far today. So we're going to